Welcome back to the Deep Dive. Today we're digging into a development in cancer research that's making some serious waves.
Hmm.
And it's not just about the science, which is groundbreaking, but there's this really powerful human element too, specifically around patient consent that makes this one feel, well, different.
You're absolutely right. Just very recently, July 16th, 2025, the National Institute of Standards and Technology, that's NIST, they, along with collaborators, released this huge public resource,
Okay.
a massive amount of genetic data, and it's focused specifically on pancreatic cancer. It's a really big step for the field.
Indeed. And significant, I think, almost feels like an understatement here, especially when you think about some of the ethical roadblocks we've seen in medical research over the years.
Definitely.
So our mission for this deep dive is to really get into why this particular data release is such a big deal. How did they put it together, and what does it actually mean going forward for research, for treatment, maybe even for the ethics around medical data?
Yeah, lots to unpack there.
Okay, let's unpack this. So kicking things off with the big news itself, July 16th, NIST and others, they unveiled this, well, extensive genomic data from a pancreatic cancer cell line. This isn't just a small snapshot, is it?
No, not at all. And extensive or comprehensive here, it really means the whole picture, the full set of genetic instructions from that specific cancer cell,
The entire genome.
entire genome, including, all the specific mutations that are believed to have caused the cancer in the first place, and the scale. We're talking several terabytes of data.
Wow, several terabytes. So, okay, you have this enormous amount of information. What's the immediate goal? What's the most crucial application right out of the gate?
Well, it's really designed as a tool for a few key groups. You've got researchers, obviously, but also clinical labs and the drug and biotech
It's up
companies.
across the board, really.
Exactly. The main idea is to give them a reliable resource to speed things up, studying cancer genes, developing potentially life-saving therapies, making diagnostic tests better, and ultimately, creating new treatments. Now, here's where it gets really interesting. And like you said, it feels different. This isn't just another data set.
Right.
This is actually the first cancer cell line that's been developed where the individual explicitly consented to making her genomic data totally public, freely available.
And that's the game changer, that explicit consent. To really grasp why that matters so much, it helps to think back, doesn't it? Many people listening will know the story of Henrietta Lacks
Hmm.
back in 1951. Her cervical cancer cells,
Yeah,
well, they were taken without her consent.
that's right.
And those cells, the HeLa cells, they became incredibly important for science, the polio vaccine, genetics, even COVID vaccines. But it created these huge, decades-long ethical and legal issues.
Yeah.
There's the famous book, the movie.
The Immortal Life of Henrietta Lacks, yeah.
Yeah.
A landmark case.
Exactly. So seeing this shift now, this proactive, explicit consent, it really does feel like turning a page, ethically, legally, maybe even culturally.
It really does. And Justin Zook, he's a biomedical engineer at NIST involved in this. He put it pretty plainly. He said, in the past, there have been controversies about using genetic data for research due to the lack of consent by patients.
Right. The shadow of the Lax case and others.
This, he says, is the first cancer cell line developed from an individual who explicitly consented to making her genomic data freely available to the public.
So it removes those potential legal fights, the ethical gray areas. Does that speed things up? Does it clear the way for faster research?
I think it absolutely does. That kind of clarity, that transparency,
Mm.
it removes a huge barrier. Researchers know the data is ethically sound, it's open, so they can spend less time worrying about, legal permissions and more time just doing the science.
Makes sense.
And there's another detail here about the donor. She was a 61-year-old pancreatic cancer patient, and she didn't just consent for her tumor cells. She also donated her non-cancerous cells, which is incredibly valuable because now researchers can do a direct comparison side by side
Yep.
to really pinpoint what's different, what makes the cancer cell tick.
That's fascinating. So you get the baseline and the cancer itself from the same person.
Precisely. It adds a whole other layer of depth to the data. So, okay, how did they actually create this data? It wasn't just one quick scan. NIST researchers used, get this, 13 distinct state-of-the-art whole genome measurement technologies.
13. Wow. Why so many?
Well, some of these methods are brand new, really cutting edge. And the thing is, each sequencing technology It has its own strengths and weaknesses. They might pick up different types of errors or be better at certain parts of the genome.
Ah, okay, so they don't all give the exact same result.
Exactly. You get slightly different readings, different potential errors. So by using 13 different ones, often from different manufacturers, and comparing the results,
You can cross-validate, find the consensus.
that's the idea. It makes the final data set incredibly robust. It's like getting 13 expert opinions on the same complex problem.
And a key feature then is that the data set includes all the separate results
Yeah.
from each technique.
Yes, that's right. So other scientists can see the raw output from each method, compare it to their own systems. It provides this really strong reference point.
We saw an image related to this work, and it kind of visualized some of the findings, showing chromosomes where bits had been swapped around, like part of chromosome 1 ending up on chromosome 2, mixed colors indicating rearrangements.
Yeah, that visualization really highlights the kind of detailed information researchers can get. Those large-scale changes, chromosomal rearrangements, they're often crucial drivers in cancer. And seeing them mapped out so clearly, validated by multiple methods, is powerful.
And this whole effort, it comes from a larger collaboration.
It does. It's part of the NIST Genome in a Bottle Consortium, which is actually a 12-year public-private academic partnership, quite
Right.
established. Their goal is basically to create reference standards for genetic analysis. This specific project falls under their cancer genome in a bottle program, which kicked off back in 2022. And the technical details, they're all published in the journal Scientific Data.
Got it. So bringing it back to the impact. For you listening, what does this actually mean in the real world? How does this data set help in the fight against cancer?
Well, let's break it down. First, for clinical laboratories, the places that actually sequence genes from patients' tumors now,
Right.
they can use this NIST database as a benchmark, a quality control check for their own equipment and processes.
Okay, so how does that work?
If a lab runs this NIST reference sample through their own sequencer and their results don't quite match the NIST data,
Uh-oh.
yeah, it's a flag. It tells them, hey, maybe our system isn't quite right, maybe we need to adjust something. It helps them ensure their results are accurate.
Which ultimately means more reliable diagnoses for patients.
Exactly. Like Zook said, labs can use NIST's database to perform quality control, and as a result, the public can have more confidence in the results produced by clinical laboratories.
That makes perfect sense. Increased confidence is huge.
Yeah.
Okay, what about for the researchers? The scientists in the lab?
For them, it's like striking gold, as you said earlier. They can dive deep into these sequences, hunt for genetic mutations, maybe find new ones, or understand known ones better. It offers fresh insights into how cancer actually starts and grows in humans.
Now, you mentioned this is pancreatic cancer data, but it's useful more broadly. How does that work? Are cancer mutations really that similar across different types?
That's a really good question. So while every cancer type and even every individual tumor is unique,
Hmm.
many of the fundamental types of mutations, like those chromosomal rearrangements we talked about, or specific types of point mutations, are actually seen across many different cancers. So even though this data is from one person with pancreatic cancer, the patterns, the types of genetic changes, and crucially, the methods used to find and analyze them are often very relevant to researchers studying, say, breast cancer or lung cancer.
So the insights and techniques are transferable, even if the specifics differ.
Precisely. It helps build a general toolkit for understanding cancer genomics.
And what about AI? Artificial intelligence is everywhere now. How does this data set play into that?
This is a really exciting area. You need good data to train AI, right? Especially in medicine.
Garbage in, garbage out.
Exactly. So this high-quality, ethically sourced, incredibly detailed data set is perfect training material. Researchers can use it to teach AI models to get much better at spotting cancer-causing mutations.
Even subtle ones?
Potentially, yes, and maybe even predict which drugs might work best against specific mutations found in a patient's tumor. This kind of gold standard data can really help push AI towards true precision medicine.
Okay, that's huge potential. And one more group, there's a tech manufacturers. The companies making the sequencing machines.
Absolutely. They benefit too. They can look closely at the results from all 13 different technologies that NIST used.
See what worked well and what didn't for each machine.
Right. They can pinpoint the strengths and weaknesses of current tech, which could lead them to improve their existing machines or even develop totally new ways to sequence DNA. It drives innovation in the tools themselves.
So it fuels the whole ecosystem, really. Labs, researchers, AI, tech development.
It really does. It's a foundational piece.
And this isn't just a one-off project, right?
Yeah.
You mentioned Justin Zook and NIST have plans for more.
That's the plan. After tackling this pancreatic cancer genome, they intend to turn their focus to other cancer types.
So we might see similar data sets for other cancers in the future.
Hopefully, yes. The long-term vision, as Zook put it, is for all this research to lead to new and better cancer diagnostics and treatments. This is step one in a longer process.
And for anyone listening who's interested, maybe a researcher or just curious, this data is actually available now.
Yes, it's freely available. Anyone can access the complete genomic data from this cell line on NIST's cancer genome in a bottle website.
Fantastic. So wrapping things up then, it feels like this NIST release is, well, more than just data. It's a real landmark moment.
I think so.
It shows how you can bring together cutting-edge science, the principle of open access, and crucially, these really important ethical considerations, especially explicit patient consent.
Mm-hmm.
And when you combine those things, you can genuinely accelerate how we understand and ultimately treat diseases like cancer.
Yeah, it creates this incredibly powerful multi-layered tool. It boosts confidence in the clinic, right?
Right. The quality control aspect.
It fuels basic research insights, helps train the next generation of AI tools, and pushes the technology companies to innovate faster. It really shows the power of doing things rigorously, but also ethically.
A real convergence. So maybe a final thought for you to mull over as we head out. As we move into a future where this kind of incredibly detailed genetic data becomes more common, what are the next steps? What ethical responsibilities do we as a society need to keep thinking about and developing to make sure these powerful resources are always used wisely, used for the greatest good?
That's the big question, isn't it? Something to keep discussing.
Definitely something to think about. Thanks for joining us on The Deep Dive.